Hello, everyone. My name is Ning Zhang from Star Wars Institute. So firstly, I would like to thank the organizers for organizing the conference and this workshop and giving me this opportunity to present my work on processing and analyzing ribosome profiling data. So this diagram compares the ribosome profiling and an RNA-seq experiment. So unlike RNA-seq reads where random fragments are sequenced. So a ribosome profiling or RiboSeek experiment sequences ribosome protected fragments. So most RiboSeek reads are in the coding regions, and RNA-seq reads tend to spread along the transcript. Also, this figure on the right summarizes what we can get from a RiboSeek experiment. For example, this green transcript forms a hairpin structure which blocks translation. And also this purple transcript has translation upstream of the classical ORF. So both of these can be discovered from the RiboSeq data. And also if we have paired RNA-seq and RiboSeq experiments, we can measure the expression from both simultaneously, and the ratio between the two reflects the translation efficiency, as is shown in this panel C. So after proper processing, a high-quality RiboSeq data should align very well between the start and the stop codon, and also present a clear 3-nucleotide periodicity. Also, typically, frame 1 should have more reads than frame 2 and 3. So however, if we plot the raw data, it looks a little bit different. So here we can still see the 3-nucleotide periodicity, but the reads are shifted upstream. So this shift is called PSI Offset. It is because when the ribosome P site is aligned to a codon, the 5' end of the read is actually upstream. And we usually only take the 5' end to represent a read for our RiboSeq data. And the PSI offset is a very important number to determine. And based on this number, the RiboSeq reads need to be shifted downstream before further analysis. So the first part of our package is data processing. We build a SnakeMake workflow for better scalability and also reproducibility. So SnakeMake creates a local kernel environment where the software packages are not affected by system-wide installations and updates. So you'll hear more about SnakeMake in the upcoming keynote talk after this. So starting from the raw reads, the workflow performs adapter trimming and read length filtering. Then the reads are first aligned to non-coding RNAs to eliminate some contaminations. And the remaining reads will be aligned to the reference genome and transcriptome. And finally, we'll also generate a summary report. So in this process, we need some user input files and parameters. And most of them only need to be set up once for the same species. So the data processing report is an R Markdown HTML file containing summary stats for each step. So for example, in this middle plot shows the non-coding RNA percentage in different samples. So the first 5 samples are RiboSeq, and the last is RNAseq. The orange bar represents the non-coding RNA percentage, and we can see that the RiboSeq data could contain many non-coding RNA contaminations. The second part of our package is data analysis and visualization. We build an R package that takes the aligned reads and a genome annotation. So here we show some analysis plots. So this is the read length plot, read length distribution. RiboSeq reads tend to be short, around 30 BP. Here are the 3 most abundant read lengths, 27, 28, and 29 BP. And also, this is a plot summarizing the genomic features that reads overlap with. And this is a metagen plot around the coding start and coding end regions. We can see a PSI offset of 12 BP, which is a very common number for many RiboSeq experiments. So the metagen plots can be drawn using all reads or reads with specific lengths, and the PSI offset might be different for different read lengths. We can also compare between the coding start and end regions. So for example, taking these two samples, the first one is a normal condition where we can see active translation. And the second one is treated with a translation inhibitor. And we can see that most reads are stacked at the coding start position. So after we select read length and also shift reads based on the PSI offset, we can then perform some further analysis. As we mentioned earlier, translation efficiency is a ratio of RiboSeq reads versus the RNAseq reads. And also from the previous metagen plots, reads tend to be high at the coding start and end positions. So usually we will trim 6 BP or 2 codons from both ends. And also we can discover new orbs from the RiboSeq data. So we implement off-score, which is a simple method to count the reads on 3 frames and test if frame 1 has the highest. As a toy example, here we show 2 orbs with 10 codons. And the off scores are identical for both because they have the same read counts along the 3 frames. But the first ORF might be more reliable because reads covers every frame 1 positions. For the second ORF, the reads only cover 1 frame 1 position. So this frame 1 coverage together with the off score can help us filter out some false positives. So our package is still undergoing development and testing. So it is available on GitHub. If you are interested, welcome to download and give it a try. And feel free to let us know if you have any questions or comments. So I would like to thank members of the computational biology. Madeline initiates the early development, and Ariel generates a lot of useful resources, and Hua gives very valuable suggestions. I would also like to thank the Bazzini lab, and Dr. Ariel Bazzini is an expert in ribosome profiling, and he provides many great feedbacks. And that's pretty much it for my talk. Thank you.